In our last video, we were talking about the adaptive immune system, and that includes the activities of the B cells, which differentiate into plasma cells and memory B cells. Now, the other part of the adaptive immune system is the T lymphocytes and their related activity. So, both T and B lymphocytes come from the same cell line. They come from the lymphoid cell line. In the process of hematopoiesis. Now, T lymphocytes, are sort of differentiate in the thymus during our, early growth and development, actually in our childhood. And similar to B cells, they have this process in which their DNA creates lots of antibodies. And each of these antibodies is going to be specific for a corresponding epitope, or part of an antigen that represents it. So we sort of are, in our early childhood, we develop lots of these with lots of different shapes to recognize proteins that have different shapes. Okay, so these all, sort of emigrate from our thymus and go into the lymph tissues throughout our body, where they sort of line the lymph tissues, and then they wait inside these huge lymph nodes, inside these lymph nodes, to be presented with their specific antigen by an antigen presenting cell, like a macrophage or a dendritic cell. And again, they have to be presented this matching antigen alongside a major histocompatibility complex that matches their own. Okay, and that's what will activate the T cells. So say we get an activated T cell here, this one comes over here, and the major histocompatibility complexes matches, and the epitope of that antigen matches, so that's like a lock and key. They fit together, so this T cell will bind here, and this T cell will become activated. Now, when these T cells, when these naive T cells become activated, they can proliferate into lots of different subtypes of T cells. They can differentiate into helper T cells. They can differentiate into regulatory T cells, or Tregs. They can differentiate into cytotoxic T cells, and they can differentiate into memory T cells. So, and there's a few others that are not, sort of talked about quite as much, but these are the major four kind of subtypes. And probably the fifth one that's the most common would be the natural killer T cell, which is not to be confused with natural killer cells. But anyways, I'm going to talk about these four today. So the T memory cell is sort of the easiest to remember, because it's or the easiest to talk about, because it's very similar to the memory B cell. This is just going to go and increase the concentration of T cells that are hanging out in the lymph nodes, waiting for future antigen presenting cells, showing them this antigen. And again, the memory T cells can then differentiate into any of these cells again, so it just sort of speeds up the recognition process for future infections. Okay, so and then cytotoxic T cells, they are called cytotoxic because they're toxic to cells, and what they do is they directly attack cells that that are infected with viruses. So this one's sort of become infected with lots of little viruses. There's a little viral capsid, and it happens to be expressing the proteins or presenting the proteins of this virus on the surface of the cell. So the cytotoxic T cell knows that there's something going wrong, so it attacks that cell and destroys it. And it also can attack and destroy other aberrant cells, cells that are showing proteins that are related to cancers as well. So cytotoxic T cells are very active in viral infections and in cancers, and in fighting off cancers. Tregs are cells that actually suppress immune responses, and I'm not going to go into a whole lot of detail. It has fairly complex activity, but it suppresses inflammation and it suppresses the B cell response. So it provides some regulation so the inflammation cannot get out of check, and it can help to prevent sepsis or a SIRS response, and it can also help to prevent some autoimmune diseases. So there is some theory that autoimmune diseases, part of the pathology or pathogenesis of autoimmune diseases is dysfunction of the Tregs. And also we know that dysfunction of Tregs is also involved in graft rejection and in graft versus host disease for patients that have had bone marrow transplants. Okay, and that's all I'm going to say about Tregs at this point. So I'm going to spend some time talking about helper T cells now. Now, helper T cells are play a very central role in our immune system, because they sort of regulate the activity of both the adaptive immune system with B cells and immunoglobulins that plasma cells produce, and they also play a very critical role in controlling the innate immune system by regulating the activity of macrophages. Now, how do they do this? Well, B cells and plasma cells, B cells will not proliferate into plasma cells, and plasma cells will not actively produce immunoglobulins in high amounts unless they are stimulated by cytokines produced by T helper cells. And again, also, macrophages are sort of wimpy and docile until they are stimulated by cytokines. So our entire, really, both branches of, these both play the central role. Without active macrophages, we have a very ineffective innate immune system, and without active B cells, we have a very ineffective adaptive immune system. So we have T cells here in the center, helper T cells, that are very critical. And why is this necessary? Why is this process necessary? Well, T cells, the helper T cells, is sort of an important mechanism to prevent our body from overreacting to perceived threats that aren't real threats. So on a T cell, we have a major histocompatibility complex paired with an antibody, and the T cell is not going to react and release cytokines unless it receives information, unless it's activated by an antigen presenting cell that has engulfed a bacteria that has this antigen, right? And also has a matching major histocompatibility complex. So this is necessary to activate the helper T cell, right? Now what happens when it's activated is it starts cranking out cytokines. Interleukin 2 is probably the most important. So what does interleukin 2 do? It stimulates macrophages to become much more active, and it stimulates B cells to actively divide and proliferate, and it stimulates plasma cells to crank out immunoglobulins. And it has a bunch of other systemic effects as well that help to stimulate, or that help to fight off infections. Like it stimulates a fever. It systemically actually stimulates some capillary leak and some sort of systemic inflammation. Now, back in the early 1980s, I should say, we began to become very aware of the importance of helper T cells because of the advent and early understanding of HIV and AIDS. So I want to talk a little bit about HIV and AIDS. So HIV, as everybody knows, I'm sure, is the human immunodeficiency virus, and that causes AIDS, which is the autoimmune deficiency virus. Now, HIV and AIDS is caused by, actually AIDS is caused by a little retrovirus called human immunodeficiency virus. And if you remember correctly, a retrovirus is a virus that has no DNA, but it has RNA. And what happens is this HIV virus has a little receptor on it called the glycoprotein G120, I believe. And glycoprotein G120 binds with CD4 receptors. And CD4 receptors, as I'm sure everybody is aware, happens to be on helper T cells. So that's why they're sometimes called helper T cells and sometimes they're called CD4 cells. And actually CD4 is just a classification of receptors that are on various cells. So there's lots of different CD cells. For instance, hematopoietic stem cells have a receptor 34, CD34 on them, so they're called CD34 cells. And then cytotoxic T cells have receptors CD8 on them, so they are called CD8 cells. So in any case, there is there's these CD4 receptors among other receptors that are, we have tons of protein receptors on every cell, and this one, the CD4 receptor just happens to bind perfectly with the glycoprotein G120, which is on the capsid of the HIV virus. So what happens is, well, here I don't want to go too far down the helper T cell role, because I want to first say that there are other cells that have CD4 on them as well, and they are the dendritic cells. And dendritic cells are phagocytic cells that are present in tissues throughout the body, similar to tissue macrophages, and they're called dendritic cells because they have these sort of dendrites that stick out of them. And so they're very active macrophage or phagocytic cells that are present in tissues throughout the body, and they have the exact same CD4 receptor as the helper T cells. Okay, so again, these are dendritic cells. And so again, the HIV virus, the glycoprotein G120 of the HIV virus binds to the dendritic cells. And it's very easy for them to gain access to the inside of these cells, because what do phagocytic cells do? Well, they phagocytize things. So this is going to phagocytize the virus and sort of engulf it and bring it into the cell, and then the rest of the story is that the virus opens up its capsid, releases its DNA along with an enzyme called reverse transcriptase, and the reverse transcriptase allows, usually DNA transcribes itself into RNA, but with the presence of reverse transcriptase, the RNA actually transcribes itself backwards into DNA, and then it inserts this DNA within the DNA of the cell, and it sort of co-opts the cell, and the cell begins producing lots and lots of HIV viruses. And eventually, after it's made many, many viruses, the cell ends up dying and lysing, and the viruses get out into the system, and then we end up with viremia, when we have millions and millions of copies of the virus running around the blood. So in the early stages of the infection, usually the theory is the first cell that gets infected are dendritic cells, and then later on, as the virus spreads throughout the body, it actually spreads through a number of tissues which also have CD4 receptors on them. It goes to the brain, it goes through the gut, and other tissues of the body. And then, there ends up being an acute reaction, acute response, which is caused by this massive viremia, and it obviously causes a flu-like symptoms. As we have a massive inflammatory response to it, and this can last for several weeks. You can also get mucocutaneous disease, which is caused by inflammation due to the virus attacking cells in the oral mucosa. You also can have nausea, vomiting, diarrhea, and anorexia for weeks, and that's also caused by viral infection and inflammation of the gut. So anyway, and then it ends up going into the body actually does very well nearly eradicating the virus, so the virus sort of has to go into hiding, and it hangs out in various tissues, but is not causing an active response. But then what happens over time is it begins to infiltrate the helper T cells and bind with the CD4 receptors of helper T cells. And then over time, the helper T cells start to die out, and we have a decreased helper T cell population. And then remember how we have lots and lots of T cells with lots and lots of antigens. What happens is we start to lose our what's called the antigenic coverage. So we have so few T cells that the helper T cells can only react to a handful of antigens, so it leaves us wide open to infections from other antigens. Now, we still can have an immune response. We still have B cells, and we still have macrophages, but these elements of the immune system are very blunted without the helper T cells cranking out cytokines. So this leaves us open to what are called opportunistic infections. Now, opportunistic infections are infections. Here, let me pick are infections from bacteria and yeast and fungi that are ubiquitous in the environment all the time. So you start to get strange infections. Like pneumocystis pneumonia, or PCP. And pneumocystis is a bacteria that's ubiquitous, but we usually have great defenses against it, so it never causes serious infection. But when you have HIV, you get this opportunistic infection. Patients with HIV can also get strange fungal infections like aspergillosis, and aspergillus is a fungi that is again ubiquitous. It's in the soil, it's in dust, it's in our houses, it's all over the place, but we don't get infected. We don't develop serious infections from it unless we are immunosuppressed. And also we can get viruses that cause certain kinds of cancers, and we also, the helper T cells play a critical role in, when we do develop cancers, fighting off the cancers, so it leaves us open to getting cancers as well. And one of the cancers that is frequently seen is Kaposi's sarcoma. Okay, and all of these infections and cancers are possible because of the loss of effectiveness of helper T cells. And essentially, without the helper T cells, again, both the adaptive and the innate immune system are ineffective. Okay. So in the next slide, we'll talk about some other pathophysiology manifestations of immune system disorders, including different types of allergic reactions and autoimmune diseases.